Alright, so today we're on to membranes. Things to remind you about. You know there's a lab exam going on this week? Yep. Anybody taking it? How was it? Easy, hard, okay? What you expected? They're giving nothing away, the rest of you. Okay. It should be okay, I think. Remember to take it. Get that done. Friday we have a test. You'd be able. Another way to say this: are you coming to class on Friday? No, do not come to class on Friday. Okay. You have that time to take the exam. That being said, the exam will open at like 12:05 or something a.m. and will close at 11:55 p.m. So you have all day to take it. But I know for a fact you have the hour that you would be in class free, so you could take it then. So no fair emailing me and being like, I have no time to take this. You do. Right? Beautiful. You'll do great. I'm going to send an email reminder out too. I realize you've heard this now, but I'll send an email reminder out as well. Should be good. Yeah? Okay, before we get on to today's official lecture, go ahead and take out your little clicker things and see if you could help me out with this concerning situation. Anybody have any personal experience with this? All right, let's see. Why does it? Okay, I apparently need to not double click. I would like to use it. Thank you. I want people to see stuff. Okay, it's not frozen. Great. No, what? Just a second. My phone is deeply confused about what's happening. There we go. Okay. Oh yeah, it's fine. How did that work for you? Oh my goodness, I know about it. Anybody I'm missing? Oh, Libby's here. Can I get yours? Yeah, that's okay. Okay. Cool. Okay, anybody who I didn't get? Yeah. Anybody else? Okay. Oh. Do you all have them? Have I asked this before? You have some? Okay. Okay. Great. Let's see what people said. Okay. People are funny. Why does the class think this is a good idea? First of all, don't microwave it, okay? Like, don't do that. Right, people think online people, the options were how to dry out a phone that gets wet. The class overwhelmingly wants to put in a bowl of rice as opposed to microwaving it or shaking it. How come? Anybody ever done this before? Hi. Yeah. What do you think about going to the rice, right? And it's not super surprising to you, hopefully, to be like, oh yeah, okay, there's some level of, if there's water in my phone and rice is dry, water's going to go into the rice, right? Okay? Intuitively, we're okay with this idea? Hopefully? Yeah. Hold that thought because it becomes important later. Another thing to look at, and I've been like all around here because again, online people, is another thing to look at. Got a glass of water here. Try to hold it as still as possible, right? Plain old boring water, nothing super exciting. And then just food coloring, because why not? Great. Can people see this okay? Yeah, it's not that exciting, but okay, if I'm not gonna like splosh stuff, I'm gonna keep it pretty still. If I let blue food dye go down one side, and red food dye down the other side, how come all that food dye is moving? The blue one's moving one way, the red one's moving the other way. How come they're moving? Yeah, it's just they're diffusing down the concentration gradients, okay? This is happening not because of some shaking motion or because one side's better than the other. It's just random movement of molecules, right? So as random movements, it's going to spread the blue out away from high concentration to low concentration, and the red out from its high concentration to low concentration, right? Just going to swoosh around. Give enough long enough to do that. This is one from the other class, right? And in an hour, it's completely sorted itself out. All right. That was, from what I remember, was where we ended on Monday. Right? We'd seen these pictures? Great. All right, so beautiful. Substances are going to diffuse or just go down their concentration gradients. All right? You don't have to, it doesn't cost anything. You don't have to pay for it. There's no work that needs to be done for this to happen. It just happens because of physical properties inherent in the universe. It's called passive transport. All right? Passive process. It's going to happen. I did this all on the board for this morning's class. I'm trying to figure out the best way to do it for this class. Dye molecules are molecules, right? They are. They're great. So dye molecules going from high concentration to low concentration, that's fine, right? Let's say you put some sugar in there. Sugar molecules going from high concentration to low concentration wouldn't shock you, right? Okay. Water is also a molecule, which you know this, right? So water goes from high concentration to low concentration as well. All right? It gets to do that. So if there's a high concentration of water in your phone, moving to a low concentration, someplace where it's dry, is not surprising. It's going down its concentration gradient. It's not costing energy, right? It's not like the rice has little wet, dry vacs that's actively sucking it out, right? Water is moving down from high concentration of water to low concentration of water. With me? When that happens, that's called osmosis, okay? So diffusion is just any solute moving around, or any particle moving around down its concentration gradient. If it's water going down its concentration gradient across some sort of membrane, that's then called osmosis, okay? We're going to see this. Can you see is this view, you guys can see okay in the back? Okay. In this first U, you've got a whole bunch of sugar molecules and a higher concentration on this side than that side, right? These little green blobs, right? Left to their own device, if the green little sugar molecules could move wherever they wanted to, where would they go? Yeah, okay, a lot of people are doing this with their head. It'd go and you'd end up with equal concentration of sugar molecules on both sides, right? Cool, not a problem, yeah? That's like our dye, it can all move anywhere, equal concentrations by the time we're done, yeah? But somebody was sneaky. We're going to actually see something very similar to this in lab next week, so, I guess spoilers for lab. Somebody put a semi-permeable membrane right here, meaning, I should turn that light off, but meaning some things can cross, but other things can't. One thing that cannot cross is sugar. Sugar, the holes are too small. So sugar is a nice chubby molecule, or this kind of sugar, I don't know what kind of sugar we're using here, let's say we're using sucrose, it can't cross. It's a nice fat molecule, it gets up to that semi-permeable membrane, and it might as well be a brick wall, right? So, too bad for sugar, has to stay on the side, can't go down its concentration gradient. Everyone's with me there? These things just happen sometimes. Okay. Now, this is a weird way to think about this. I realize it's a weird way to think about it, but there's a higher concentration of sugar over here, lower concentration of sugar here. Is everyone okay that concentration is the amount over how much area you have? Right, it's not just total amount, but it's amount relative to the area. Okay, so if you have 2 pet chipmunks in your dorm room, right, you have a higher concentration of chipmunks in your dorm room than, say, I don't know, this whole building. That maybe there's 3 chipmunks in this whole building, but there's a lot more space. But there's less space in your room, so there's a higher concentration of them. You okay with the idea? Usually in textbooks they're drawn equal sizes, just so you're aware that it's all the same. But it's not just a number; it's a number based on space. But okay. Higher concentration of sugar molecules over here, lower concentration of sugar molecules over there. Great. Straightforward. But remember that water is a molecule too? Where is there a higher concentration of water molecules? Is there a higher concentration of water molecules on this side or on that side? Which is this side or that side? There's more water molecules over here, right? How come? Wait. But the level of water is the same, like equally high. How come Ben, he's right, but how come you're saying that there's more water molecules on this side?
Because the sugar is taking up space also.
Yeah, so I'm going to repeat, not to be annoying, but because the sugar is taking up space as well, so there's not as much water there. Okay? Everyone's with me there? So then, if there's a higher concentration of water on this side than there is on that side, where's water going to go? Because water can cross that membrane. Water can cross, sugar can't, but water can. So water's going to go down its concentration gradient. Where does it go? Yeah, so water's going to push, and water is going to keep going until it's like, if this is some like weirdly high thing, until gravity overcomes it. But it's going to be pushed until it has similar concentrations of solute, right? And so now at this point, per unit space, things are equally concentrated. There's equal concentrations of water on both sides, equal concentrations of now this solute, the sugar on both sides. Does that make sense? We're okay with this? I feel like there's a question there. No? Yes? This one, there's more physical water on this side, but the concentration is the same. Yeah? This is that same picture, but now we're actually seeing the water molecules as well. And so this is osmosis, this diffusion of water molecules down their concentration gradient across a semi-permeable membrane. All right? Beautiful. That sets up some other things. So most cell membranes, water can cross, all right? Because there's aquaforms. So water can cross. But a lot of other things, a lot of proteins, they might be there in the solution, but they can't cross either. Okay? So I have things to show you. But I'm here as well. Okay. I have a little egg, right? This is an egg-sized egg. It's like a normal-sized egg. Okay? It's actually like jumbo-sized egg from the store. Okay? Beautiful. I deshelled a bunch of eggs with vinegar the other day, so the membrane's still intact, but the shell's gone. Okay? And then this little guy. Notice any subtle differences in size? This one's a big fat egg, right? Okay. It started the same size as this one. Okay? But now it's big and fat. There's a bunch of chunks inside an egg, right? An egg works as a cell. Lots of chunks, lots of proteins, right? Some sugars, some other things, okay? Lots of stuff inside the cell. Most of that stuff cannot cross the membrane. right? So I got this little egg, it was smaller, I plopped it in just a bowl of water, and now it's fat. What, what happened? Water went in, and how come water went in? Yeah, there's a lower concentration of water inside, right, relative to the pure water concentration outside, and there's a whole bunch of solute chunks inside, right? So water poured in, right? I mean, it took a little while, but water came in and it got really big. Fair enough? Okay. This egg is kind of cool. Have you ever done this before with eggs? Oh, you should totally try. I'll leave some in the lab for people to play with. This egg was once like this little guy, right? Egg size. Notice any subtle differences in like size? It's little and it's kind of mushy, right? Um, the membrane's still intact, right? Like it's fine, I can hold it. This one I put in sugar water, right? Lots and lots of chunks of sugar, right? My little egg, which originally was big, how come it got little bitty? Water left it, right? So if there's more solute chunks outside relative to inside, the water's going to leave, right? Until it's got equal concentrations on both sides, right? We're solid on this? Okay. Water doesn't know about egg protein, so it's not like I need equal concentrations of egg proteins on each side. It just knows that there's a lot more, that the concentration of water outside is lower than inside, so it's going to leave until the water concentration is the same. Fair enough? Because this is biology, there are names for all of those things. You know them intuitively, right? But there are names for them. Oh, one quick question. If I wanted to have an egg stay exactly the same size, because like I care deeply about the egg being the exact same size, if I want to put it in a bowl of something, do I need to have that bowl of, it's going to be water, do I have more solutes outside, less solutes? How do I make it stay the exact same size? Same. I need to have the same concentration outside as inside, right? So like, the really easy way to do this would be if I cracked a bunch of eggs, right, and put one unshelled eggs and a whole bunch of like egg whites and egg yolks. Nothing's going to change because the concentration outside and inside is the same, just sit there, right? Okay, so terms. Describing the overall situation, just how is the surrounding solution, how chunky is it, is called tonicity. So it's called you can use the word tonic to describe a situation, a solution. So an isotonic solution is one that the concentration of solutes on the outside and on the inside of the cell, it's the same. So there's no net movement of water across the membrane. All right? Water's still going to go back and forth, right? Just because of random motion. But the cell's not going to gain water, it's not going to lose water, it's going to stay the same. All right? When somebody's hyperactive, are they overactive or underactive? Over, right? Okay. So are we okay with the idea of hypertonic? There's going to be more solute concentration. There's going to be more chunks relative to the inside the cell. A hypertonic solution has a lot of solute chunks. All right. And because something's hypertonic, it's going to have the cell lose water. So our little egg, right, in a solution that has a lot of solute chunks, water's going to come out. The cell loses water. Hypotonic is the other way. So like hypothermia is not enough heat, right? Hypotonic is there's not that many the solute concentration is not that high in the solution relative to the cell. That means the cell's going to gain water, right? Water's going to come in. Yeah? So far, so good? And depending how you word this, so like, this cell is hypertonic relative to the solution, and the solution is hypotonic relative to the cell. Okay? It's just you're saying which one has more chunks. Yeah? Interesting things happen because of how this works, because of some things able to cross the membrane but not everything. It sets up interesting cellular properties. This is more if you're studying on your own. Okay, I need to show you pictures. And I will then say the words on the slides. Pictures. If you've got an animal cell, say red blood cell, beautiful. It's in an isotonic solution, like inside your body. It's not going to gain water, it's not going to lose water, it's going to do its just normal blood thing. Okay? Great. If you put those same cells into, say, pure water, relative to a blood cell, it has all sorts of chunks, it's got proteins, it's got all sorts of junk inside of it. Relative to, well, is water hypertonic, isotonic, or hypotonic? So basically the question is, are there more or less solutes in water than in pure water than in a blood cell? Less, right? So it's hypotonic. Okay. So if it's now in a hypotonic solution, water's going to go down its concentration gradient, right? Lots of water here, not very much water inside because there's so much other stuff taking up space. It's going to come in, right? And the cell will get bigger, and the cell will get bigger, and it'll pop. So don't use water as a replacement for blood transfusions. Don't do that. Okay? Right? Everyone's good here? This is why people need to, like, hydrate somebody, right? So if someone's dehydrated, and they run, like, an IV in on people, they don't run pure water in. They're like, well, that's the fastest way, because then you just get water. No, you run in saline, right? Because you don't want it to just, like, pop things, all right? Saline has solutes in it. It's going to prevent this kind of thing from happening. Right. Okay, cool. The opposite thing can happen, too. If you're hypertonic, so you put the cell in something where there's a whole bunch of solutes outside, the cell loses water and it's going to shrivel. All right? That's kind of bad news. Plants actually don't have such a problem with hypertonic solutions. We're going to look at all three of these. Has anybody, like, ever decided to be super healthy and you get, like, a whole bunch of celery? You're like, yes, this is my new, I don't eat so many gummy bears, but celery is for me, right? Okay. And let's say you maybe, for some reason, forgot your package of celery in the bottom of your fridge, and, like, you come back eventually to it. Is the celery nice and crisp? What has happened to it? It just goes limp, right? Like this is pre-rotting stage. Let's assume you don't let food rot in your refrigerator because, like, who would do that? Gosh, good grief. But it just goes limp, right? Okay. So, plants and bacteria and some other things have cell walls, okay? It's this rigid-ish structure outside of the actual cell. So, if you have the plasma membrane here inside the cell, and it's in some sort of hypertonic situation, meaning water is going to leave, right? Water leaves. The cell membrane, plasma membrane, pulls away from the cell wall because it's shrinking, right? And makes the whole thing limp, right? It's called plasmolized, which is a cool word, but just right? Okay. This is very bad for plant cells, right? Like that. Also, even if it's in if it's in some sort of isotonic solution, it's still fairly squishy because it's not pushing up against the cell wall, right? But if you have a plant cell in, say, a hypotonic solution, say water comes in, right, and the cell membrane puffs up, right, because water's going down, its concentration gradient comes in, puffs up. It pushes up against that cell membrane, that cell wall, and the cell wall pushes back, right, once it's held tight. You can imagine, like, blowing up a balloon inside a birdcage, right? As you blow, the birdcage pushes back. Does that make sense? This is the same reason why, when you go to grocery stores, they'll, like, mist their fruits and vegetables, right? It's not to, like, keep them fresh longer. They're cold, like, they'll be fine. It's because if you put water on it, those carrots will take it up, right? They'll be stiffer. They'll look fresher. I mean, they'll do better, right? They're crunchy. Fair enough? Um, questions about this? Beautiful. There's so much to talk to you about. But people are nodding at least, so that's good. I feel like people are kind of with me here. All this we've talked about, osmosis is a form of diffusion, all this just down concentration gradients, passive processes. You can have facilitated diffusion. Water typically is facilitated diffusion. It's just when this diffusion is happening because you've got some channel proteins that let this happen. Or carrier proteins. So here's the picture. This still counts as diffusion because there's a higher concentration here, lower concentration here. Down is concentration gradient. Yeah, it can't cross this membrane by itself, but the membrane has proteins in it that let it cross. We're okay with this? Yeah? Still a passive process. You don't have to pay any energy to get this done. It's beautiful. But okay, so maybe, like, this is all great. Life's good. But maybe you're some sort of cell that wants to live in a hypertonic environment, but you just wants to, like, spend your life pumping things against their concentration gradients. You can do that, right? That's okay. If you weren't here on Monday, this was an extended analogy for Monday. If you've got a whole, like, room full of toddlers, right? And they're just spreading out, you open up the room, they're down the concentration gradients, they're spread out as possible, right? But you have this, like, improving book on, like, being friends or something that you've decided you want to read to them, right? Have you ever tried this? You can physically get it done. You can be like, okay, kids, we're gonna read this book on the importance of sharing, right? Does it cost you anything to have this happen? Like, expenditure of energy. There's, like, a bunch of these kids, they're running everywhere. Whew. You're gonna have to do something to make this happen, right? Like, you have to pay something, okay? You can do it, but it's gonna cost you, alright? The cell has a similar idea. Cells can move solutes against their gradients. They can move solutes ways they wouldn't go just on their own, but the cell has to pay for that, alright? This is called active transport, where the cell's actively transporting stuff, as opposed to just, like, letting it move. Yeah? Concerns about this? It's great. You need energy. The energy is typically ATP. And I have pictures for you. One of my personal favorite examples of this is the sodium-potassium pump. It happens a lot in the body. It happens for neurons. Your brain hogs down a ton of energy at rest, like a lot. And most of that is going to just powering this pump. Because for reasons that you don't care about right now, but for reasons, neurons really need the inside of the cell to have high concentrations of sodium, low concentrations of sodium, high concentrations of potassium. Okay? But left to its own device, we've got high sodium outside, low sodium inside. Sodium just left to diffuse, where would it go? Inside. It desperately wants to go inside. There's a whole bunch of sodium outside, not very much inside. It would love to come in, right? And potassium's the other way around. Potassium would love to leave, right? But the cell wants it to not be this way. So the cell spends a lot of energy actively pumping sodium out and pumping potassium in. And the thing is, these things leak across other channels. And so the cell is basically always shoveling sodium out and potassium in. It just doesn't stop. If it stops, you die. Sorry, it sounds like a threat. I don't mean to sound... Many neurotoxins act on this. That's a better way to say that. Okay, so this pump takes in sodium, it costs ATP, it changes in shape, spits sodium out, takes in potassium comes in, and it just keeps going. It's pulling potassium against its concentration gradient, pushing sodium against its concentration gradient, but because it paid for it with energy, it can do that. All right? Good? Yeah? Active transport, moving things across membrane, but this time you've got energy and you can go against the concentration gradient. So here's the overview, right? This is an example of active transport. It's energy here. You'll notice moving against. Passive transport, you can have something like just diffusion just happening across the membrane proper, or facilitated diffusion through some sort of channel protein. Yeah? Everyone's good with this? Okay. One of my favorite things about this, I don't know why, I just think it's kind of cute, it's called co-transport. And that's when the active transport of one thing drives something else to happen. If you're, like, at least a little bit intuitively aware of this, anybody in this class, when you're about 16, if you got a driver's license, it was great, and then you could borrow the car all the time, but you also suddenly became the family's errand person? Did this happen to anybody else? Like, great, you can drive also. Get milk, pick up your little brother, and we need, like, toilet paper. You're like, oh, okay, right? Does this experience ring true to anybody? Okay. So, what that's basically saying, with this dumb analogy, is, yeah, something that is energetically favorable for you, having the car, is coupled to something that you basically have to pay for. You have to go pick up milk, okay? In the cell, this happens a lot. The active transport of one thing is going to drive the transport of another thing. So the example would be here. Sucrose, as you know from lab, is fructose and glucose stuck together, right? If you throw acid on, you can snap them apart. You saw this, right? Beautiful. Okay, sucrose is this big fat molecule, and you want to take it in. Right, you want in the cell. The cell's very happy to chew up sucrose. It loves sucrose. There's already probably sucrose inside the cell. So sucrose, because of the amount, the concentration, and because of how big it is, it's not just diffusing across the membrane, right? You have to bring it in, all right? Well, this would cost something, and so the way for this to come through, the cell basically says, you know what? You can bring sucrose in, but bring hydrogen with you, all right? So hydrogen, there's a really, really high concentration of hydrogen on the outside. So hydrogen wants to fall into the cell. Hydrogen wants to diffuse down. So hydrogen diffusing down, whee! As it diffuses down, it's going to open up a spot for sucrose to come in as well. We're okay with that? The cell's paying for this by this hydrogen going down its concentration gradient, right? The way the cell actually sets up the concentration gradient is active transport. It's actively pumping hydrogens out, pumps them out, and like as soon as it gets out, they're back in. Whee! We're okay with this? This is called co-transport. It's active transport of one thing is indirectly pushing something else around. Yeah? Again, takes energy, but we're there. Okay? So I know we've seen a lot today. We're about to see more. But we've seen, okay, just molecules move, right? Molecules move randomly. Things go down their concentration gradients, right? If they can cross the barrier. If they can't cross the barrier, they're not moving, right? Water goes down its concentration gradient, right? Osmosis, all right? And then depending on relative chunks to what's in the cell, that can make a cell swell, it can make a cell shrink, right? The tonicity question, I will totally put some question about tonicity on the exam, okay? We're solved there? Great. But this is all beautiful, you've got a membrane, you're moving stuff back and forth. Cool. What happens if you want to move something honkin' huge into a cell, right? Like sugar's big, but it's not like bacteria size, right? There's no way to get a big enough protein there in the membrane to actually move something that big through, right? I mean, I guess theoretically you could, but that'd be like your whole cell. That's a problem. Or maybe you don't want to just be like picking one or two hydrogens up at a time. You want a whole big glob of them. you're going to need something else. All right, so we have transport across the membrane. We want now to move on to bulk transport, where you're moving things in a big wad. All right? Fair enough? Small molecules, you don't usually do this too. Small molecules just enter or leave through the bilayer or through channel proteins. This could be passive with diffusion, or it could be active with active transport. But if you're going to get something really big, say like a protein across, you're going to need to use bulk transport. Bulk transport takes energy. And you know this. If you're like bulk transporting because you decided to help somebody move, right? Yeah, people who move, like when you move, you're super organized. But when other people move, they've got like boxes full of bowling balls, and you're like dragging them around because they're noble. Great. It takes energy. You know this, right? It costs energy. The cell's the same. If you're gonna have bulk transport in the cell, it's gonna cost energy. I have a boring video for you. It's this one here on YouTube, people. It's informative, it's just not super exciting. Here we go. And then we'll talk about this whole thing. Alright? Cell, nucleus here, right? Okay.
By single-celled organisms are often
Can you hear it okay?
particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein cladrine coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
That's okay? Yeah? Have you heard about this before? Huh, well, good. I'm glad. It's just apparently you're cool with it. Alright, so I'm going to say the exact same things the video just said, but more. Exocytosis, things are exiting the cell, okay? Instead of just having like things diffuse across one at a time, you've got a big vesicle, a big bag of this stuff, you bring it to the cell wall, you open it up, and you're having these things exit the cell. Exocytosis, okay? Endocytosis is going to be forming vesicles and bringing things into the cell. They're going to be entering. And like the video said, there are three types. So we've got phagocytosis, where it's basically eating something, yum. It's getting around something big, all right? Pinocytosis, you can think of the cell drinking or gulping, because there's a whole bunch of stuff here in solution. The cell is going to pinch off and get this blob of fluid, right? And then receptor-mediated endocytosis. All of these are types of endocytosis, by the way. You have these little receptors on the cell membrane, and once they're full, basically, once something is stuck to them, then they gulp off into the middle. So for phagocytosis, here's actually a real picture for it. You see this little bacteria, buddy? Yeah, the amoeba wants to eat it, right? Bacteria is big. The amoeba is stretching its membrane up around it. It's gonna swallow this bacteria, right? Yum. Fair enough. You'll note how this side right here, the side membrane that's facing the cytosol, is still facing the cytosol here. And this side membrane that's facing outside is still not touching the cytosol, right? The faces are conserved. Great, so that's phagocytosis. The immune system will do this. Like, you've got cells that can go chew up foreign invaders, right? Phagocytosis. Pinocytosis is this one where it gulps extracellular fluid. You can see it happening here, right? You've got the edge of the cell, right? The plasma membrane. You can see there's some fluid or something that they want. It gets deeper and deeper and then pinches off. See that okay? And then receptor-mediated endocytosis is very, very similar to pinocytosis, but you've got all these nice little, receptor proteins there. You can see it. You can see the best. You can see the ligand. You can see the stuff there, and it pinches off inside. Yeah. So far, so good. Questions about that? Okay, so the things to think about today were what kind of transport cells use, right? Whether it's active or passive, whether it's diffusion, right? Things to think about. We now have about 5 minutes. Oh man, I talked faster than I did before class. We have about 9 minutes. Do people have questions about the exam? What's on it? How to study for it? Okay. What's on it? How to study for it? All things. Okay. What's on it? I have to write it. I will be doing that later today. I'll also send you an email. If you have a question that you're like, this question is so beautiful, it encapsulates all of biology into this one beautiful question, feel free to email them to me. I'll try to put a couple of them on the exam. And they'll be like, I wrote that and I know the answer. Okay? If you don't want to, you don't have to. But if you want to, go for it. But you all got a study guide I sent? Yes? Hopefully? Okay. Study guide. I'm going to use that study guide and then be like, I said there'd be like, about 8 questions. I'm going to pull questions from it. Okay? The order might not be exactly the same, so I might like shuffle the order, but it's going to be from the study guide, okay? Not word for it. If the study guide's like, what's endocytosis? There might be a picture and like, what cellular process is happening here? You're like, oh, endocytosis, right? It's going to be quite a few multiple choice, quite a few short answer. There'll be a couple essay questions, right? It's timed at an hour. I'm going to try very hard. I will try very hard to give you enough time to do it, right? To not have like a million essay questions, but not so much time that if you've never seen any of these words before, you can just teach yourself all the biology you need to know in like the hour, okay? So like, yes, it's open book, it's open note, it's even technically open internet, so I know that feels weird. It feels weird to me too, like it feels weird. That's not cheating, so don't feel bad. What is cheating is, say, emailing your molecular biologist cousin. That's cheating. Having somebody else in the class take it for you, also cheating. I will check, like I can't actually check that much, but I can check answers and times on things. So don't cheat. Because that's bad for everybody. But yeah, you can do it. An hour should be enough. You do not have to take it during lab time, but you definitely have to take, during class time, you definitely have to take it that day. And I will be cold-hearted, super cold-hearted to anybody who's like, I forgot. You didn't have class. You could take it during class, okay? Yeah. No, okay, so it's sad this semester because, like, I'm not supposed to reopen quizzes typically for, like, reasons. But I feel mean being like, haha, I feel mean. Other things? Did people look at the study guide? Anything that, like, threw people off deeply? Let's look at it briefly. What? I know it's a public computer. Let me off. Well, maybe we won't. Let's try this again here. That was another exam I was giving. I can answer direct questions if you have them about stuff. Yes, it's on Almet. Okay, apparently it doesn't want me to be on this computer. It's going to be an Almet. One of the best ways to study for it, though, is going over your Mastering Biology homework, probably. Yeah. Yeah? Sweet. That's it. This is not super important, but I will leave the squishy eggs in lab, so next week when you're in lab, you can, like, squish the eggs. Right? Okay, maybe you don't want to. I was showing the other biologists, I had these, I was carrying them out, and my boss walked past, and I was like, do you want to see a squishy egg? He's like, I'm okay. Fine. Anybody who didn't put the little clicker thing up? Who are you? Okay.
I came in late because I was taking a test.
Oh, nice. And Corinne as well? Anybody else? Yeah.
Who that is.
Oh, nice.
They dropped it.
Sweet. She wants it. I'm sure she wants it back. That reminds me, was it you got keys?
Yes, I got
Okay,
them.
it was...
I came back. I brought them.
Thank you.
I
Gotcha.
remember today.
Thanks, it makes me deeply happy. I appreciate it. I'll put them in my office. Though I'm taking one home for my kid.
Okay.
Yeah,
I came back and they weren't in here and I was like, oh no, but my roommate was with me.
Oh, okay.
And she works in the library, so she just went up there and asked and they have them.
Yeah, okay. Yeah. Perfect. Oh, I feel so loved. Great, yeah. I thought the yours
I don't know where her office is.
Yeah, no, you're good. Yeah. Yes.
I don't have the...
Oh, right.
How dare you?
I remember specifically, it was on my desk.
Yeah,
And then
I think I got both of you. Gotcha, okay, thanks. I just needed, like,
I actually
hands.
brought mine this time and then she did it at the beginning. So I was like,
I was mixing things up.
I was on a very clear vision of seeing it on my desk and moving it, but not paying